In this video, we will discuss about inflammatory response that are induced by ER stress. So, whenever there is a ER stress, we know that from our previous videos, three kind of signaling pathway gets activated. First one is ATF6, second one is PERK pathway, another one is IRE1 alpha pathway. So, we will start with the PERK pathway. So, whenever the ER stress is there, this PERK pathway gets activated, and this leads to the phosphorylation of EIF2 alpha factor. And what it will does is, one function of this EIF2 alpha is to attenuate the production of IK beta. And when this IK beta production reduced, it will no longer be the ratio of NK beta and IK beta will increase. This leads to the more NK nuclear factor kappa beta in a free condition, and more nuclear factor kappa beta in the free condition will leads to transport of this nuclear factor kappa beta into the nucleus, and then activate the inflammatory genes. So and other function of this EIF2 alpha is to uh, attenuate this, or we can say modify this ATF4 mRNA in a form that it will make it ATF4, and that will go into the job, that will increase the formation of job protein. And what this job protein does is, job protein helps to reduce the attenuation through two ways. First one is that it will decrease the uh, number of free nuclear factor kappa beta, and also it will uh, make degrade this, or make this JNK factor dysfunctional. So this is how this PERK pathway plays a role in inflammatory response. So, and let us first discuss this IRE1 alpha. What it will does is, it will, whenever the IRE1 alpha signal is activated because of ER stress, it will interact with TRAF2, and that will interact with JNK and IKK. So first we will discuss the IKK part. The IKK part does is, phosphorylation of IKK will leads to phosphorylation of IKK will leads to more formation of this nuclear factor kappa beta, and this will lead to inflammatory gene response. So this IKK helps in uh, IKK helps in, we can say, reducing this IK beta, so that will lead to more number of nuclear factor kappa beta. And other ways, uh, other method by which IRE1 alpha affects or induce inflammatory response is through phosphorylation of JNK signaling pathway. So it will lead to the phosphorylation of AP1, and this will lead to uh, upregulation of the inflammatory genes related to JNK pathway. Next, or the third signal, is the ATF6. So what happens in this case, whenever the ER stress is there, ATF6 will go to the Golgi, and where the regulated proteolysis of this ATF6 occurred, and it will cleave into two sites, the homodimer, and these sites will form a homodimer, and then will move into the nucleus, and that will activate the acute phase response gene. So this also contribute into the inflammatory response. This is how the ER stress will cause the inflammatory response, and what are the other ways that this job will try to reduce the inflammatory response by reducing the free nuclear factor kappa beta and JNK phosphorylation pathway, and simultaneously it will also triggers IL-23, that will also helps in reducing the inflammatory response. So if you need to understand this inflammatory response and its relationship with ER stress in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.